Okay. Thank you, everyone, and sorry for struggling few technical problems, but I'm glad I'm here, and also it's my pleasure to share my thoughts with you. So I was told that in this session, we're going to mainly talk about two subjects. One is to update about Charcot-Marie-Tooth type 4, and then also share update about the information of related to hereditary neuropathy with liability to pressure palsies, abbreviated as HNPP. So we will first talk about the cause for these different diseases, and the second we will describe what is the approach in terms developing therapeutic uh, um, method against these diseases, and then also update about uh, the therapeutic project and our partnership with pharmaceutical companies. So next slide. So what is CMT type 4? So before I talk about CMT type 4, and I want to introduce the concept of autosomal recessive mutations. And so as you guys see on the screen, that here's a cell. Every human being has all kinds of cells. And inside those cells, there's a chromosome, which is made by the DNA. And then if you zoom in and look at a small area of this chromosome, and you will be able to identify individual genes. Each gene have two copies, one copy here, one copy from here. And one copy supposed to be from mom, and one copy will inherit it from your father. If there's a disease that only happen if there are mutation in both copy of the genes, then we call this called autosomal recessive. If the disease can be caused by by mutation in only one of the copy of the gene, one of them, not two, only one, then it's called autosomal recessive, autosomal dominant. So what we're talking about here, can we go back to the previous slides, please? Yeah. So what we are talking about here is the disease that are caused by mutation in both copies, not just one copy. So it's called autosomal recessive. There are many different subtypes of autosomal mutation that can cause different type of CMT type of 4. So first of all, any CMT, if it is caused by autosomal recessive mutations, we classify them into CMT type 4. Okay, and then under the CMT type 4, we divide them into different subtypes. And for example, CMT type 4A caused by mutations in both copies of this gene called GDAP1. And our discussion, so there's so many of the CMT type 4 subtypes. There's no way we're going to cover all of them. And I am going to focus on this one, CMT4J, which is caused by autosomal mutations in both copies of a gene called FIG4. Next slide. All right, so when the patient has autosomal recessive mutation in CMT type 4, and clinically, they will present with sensory deficits. By the way, that all autosomal recessive type have similar clinical presentation. And when they damage the sensory nerve, and then the patient will feel that they lost the sensation on their feet and hands, and they will have difficult to maybe feel the temperature when you go to the take a shower and don't feel temperature very well in the water, or you got poked on your feet and you don't feel much pain, and those cause sensory loss. Or instead, you could have increased the sensation, and sensitization means that even something that touch you that not necessarily cause pain, but they end up cause pain. And so that's also the sensory problems. So either too sensitive or lost the sensitivity of the sensory function. And there's also problem with motor function, which means that you will get a weakness in your ankle, you tripping, and or you have trouble with your hand dexterity, or trouble walking, and time goes along, it's getting worse. And in most severely affected patient may end up in use a walker or wheelchair. And in this disease also could affect the cranial nerves in some patients, which means that could have hearing problems. Occasionally, some CMT type 4 may also affect the swollen function, which they feel like frequently food stuck in their throat, or aspirate. But these are infrequent problem. And in most of the CMT type 4 do not have problem with your central nervous system, but in some CMT type 4, such as CMT type 4J, and they may end up have some central nervous system problem, which means the patient may have, the child may have a mental retardation, or have seizures, or there's a developmental problem, means the brain did not develop normally. And because this disease, they produce some kind of pathology. And so this is what the normal nerve looks like. And this dark circle, that's the myelin. In the middle of those opaque area, that's the area where we call axon. Okay. And then some of the CMT type 4 may predominantly damage this dark part, the myelin part, and we call demyelination. And some of them may predominantly damage the axon, and we call axonal degeneration. And the therapy, whatever therapy we design, we aim to either stop or reverse the demyelination, stop or reverse the axonal degeneration. Okay, next slide. So CMT type 4, as I just introduced, is autosomal recessive disease. Again, which means the disease will be caused by mutation in both copies of the gene. In other words, that if this gene, one of the gene here, one copy, and then the other copy, one from mom, one from dad, that this disease only occur if the mutation affect both copy of the gene. Most of the CMT type 4J and many other CMT type 4 also same thing. They caused by mutation, what we term called compound heterozygous mutation. What does that mean? Means one of the copy may caused by some kind of mutation, say one of them called frame shift mutation, and then the other copy may caused by different type of mutation. And not necessarily has to be frame shift, it will be something different. So that means that each of these copy got mutated, but they mutated in different way. That's why I call compound heterozygous mutation. So what does the frame shift mutation is? Basically, it means that this is a normal codon like this, and suddenly, due to whatever reason, they introduce some extra nucleotide or missing one extra nucleotide. So all these subsequent codon, downstream codons, will get altered, and then coding amino acid for the proteins also altered, so that everything below the amino acid is end up wrong, translated wrong. So that's called frame shift mutation. Alternatively, if there's something nucleotides got mutated and create a premature codon here and stop the translation of the gene, and then everything below this got stopped, and then we call this called truncation mutation. All this mutation did not affect, did not do the frame shift, did not do the truncation. Instead, they only affect the individual codon and turn the normal amino acid to something else different. That's called missense mutation. Okay, so despite it doesn't matter what kind of mutation is. Usually, for autosomal recessive disease, the mutation in two different copies end up different. So that's why they call compound heterozygous mutation. So why I'm introducing those concepts? Because the way to treat the disease, uh, you have to tailor your treatment strategy based on different kind of mutations. Next slide. So these slides, I want to introduce a very important concept in order to understand how we develop treatment for CMT type 4. That is loss of function. So as you see in the previous slides, they both gene copies got mutation, and if each of those genetic mutation of each copy, and that mutation end up make the gene does not produce any protein. It did not produce on the first one. It did not produce the protein from the second one. So that gene end up produce no proteins. That means we don't have that protein expressed, or that protein amount of that protein significantly reduced. So that means that protein function is gone. So we call loss of function of that protein, or loss of function of that gene. So because that function is gone, so it is logical to fix that problem by replace that protein got lost. So the typical example is the Pompe's disease, which is inherited disease that affect the muscle mainly. But for many years ago, and people end up produce the protein, whatever the protein get lost, and infuse it back to the patient and make patient better. So that's the protein replacement therapy for recessive disease. And so that's a good example and has been quite successful. But there's some caveats for that, because when you produce the protein and then you infuse that protein back to the patient, and sometimes you may cause the allergic reaction or some unexpected reactions. And sometimes even you produce the right protein, you give back to the patient, the protein may not be able to arrive to the specific cell or to the area that protein is needed. And then that treatment will not successful either. Or you produce a protein, you give it back, but then your body develop a mechanism and quickly degrade that protein. So that means the protein have not get a chance to arrive to the right place, already digested, and then that's not going to work either. So for that reason, and there are many other therapeutic methods have been devised to treat these kind of diseases. So the other strategy is simply replace the gene. So that typical example for that is the spinal muscular atrophy, which due to a loss of gene function called SMN. And then in the past few years, there's a major advance on this disease where people devised a virus called adenovirus. They will put this gene, that missing gene, in the virus, and the virus carry that gene back into the human cell. And in this disease, it's back to the neuron. And then because that gene got back to the cell and produced that protein, which is missing in this patient, and then disease gets better. And this has been one of the major breakthrough in the field, and really made a huge difference for those individual patients who have SMA. Now, again, just like everything in the world, and everything has their shortcomings and their strength. But in this therapeutic approach, there's several caveats. One is that viral vector is very difficult to be produced. It takes a long time and to produce adequate amount of the viral vector to treat the patient. So that is a huge roadblock. Second, there is potential that this virus vector goes into the human cell, may hit on the wrong spot. That's called off-target effect. That could be a potential another problem. The third potential issue is the gene that under the vector that produced did not go to the right cell you want. Say that disease affect glial cells as a major target, but then your viral vector produced did not go to the glial cell, then it would not be enough to cure the disease. So those are the potential caveats. And then the next strategy is called DNA bandage. So the typical example of those is the success in the Batten disease, which is a disease that causes lots of seizures in babies. And so what this strategy does is to produce a small nucleotide, which is basically a whole bunch of them. And that means usually 10 to 12 of these nucleotides that connects together that match the area of the mutation, and then patch the where that has mutation, and made that gene who will not be able to produce a protein now is able to produce a protein, and that's why I call DNA bandage, and then try to use that strategy to cure the disease. And turns out this strategy was successful in some of the disease. The advantage for this one is there's no problem for production. You can produce anti-oligo nucleotides very quickly. And also that these, because it's a small molecule, you can penetrate to all kinds of cells. For each injection that these anti-oligo could stay in human body for 2 months or even longer. And so all these are advantage of this, but there's also have caveats. The caveat is that it's not a permanent fix, so you have to repeat these treatment every few months. That's probably the main the shortcoming of this. And then in terms of CMT4J, I already mentioned about the clinical presentation. It's caused by recessive mutation. There have been animal model being produced that recapitulate the pathology of these diseases. And then over the past several years, there have been gene therapy devised against this disease, which many use adenovirus to carry the gene called FIG4 back to the neurons and other cells, hopefully, and to replace that missing gene. And this strategy turns out to be very helpful, at least successful in animal model of CMT4J. And then translation to the humans, now in the stage of interaction with FDA, and they also FDA wants require this therapeutic development team to do more work on the natural history studies, and then probably also the better outcome measurement needs to be developed. And after that, hopefully that gene therapy will eventually get translated to the clinical side. And we still have some way to go, but it's getting closer. And this work mainly gets done in the partnership with a company called Neurogene. Next. Okay, now there's a few more minutes left, and I'm going to switch a gear. It's a new topic that are going to focus on the disease called hereditary neuropathy with non-ability to pressure pulses, abbreviated HNPP. And this disease is very close to my heart. I've been studying this ever since 2002. My laboratory has been continually funded by NIH to study this disease. And I started this work when I was a junior faculty at Wayne State University. And then when I moved to the Vanderbilt for another 9 years, I've been studying this disease. And after I returned to the Wayne State University in 2018. I'm still continue to study this disease. So I have been studying this for a long, long time. So this disease is very special. In my opinion, does not belong to any classification of any CMT. Clinically, and this patient will present with sensory problems or muscle weakness or atrophy of the muscle. It means that there's a nerve damage affect the sensory nerve, affect the motor nerve. But the way they affect them is quite unusual. They in at least in the early stage of the disease, they tend to affect them in the focal area. So then these patients, their nerve is sensitive to the mechanical pressure. So for example, if a patient sitting somewhere cross their legs to talk to someone, and one of the nerve in their leg called peroneal nerve gets compressed, and then when the patient stand up, they may end up have a weak ankle in that leg. So it's very focal area. Or sensory nerve, and I had a young girl see me, and she go to school every day with a the book bag hang on her right thigh. When she walk, that book bag bounce back and forth against her lateral thigh on the right side, and then a few months later, she ended up have a sensory loss, the patchy sensory loss on the side of her right thigh. So that kind of mechanical sensitivity and those mechanical pressure on the ordinary people may not cause a problem, but they do cause problem on the patient with HNPP. Time goes long, in their later stage of this patient, they may end up have problems that affect both feet and hands on the both sides, and then became more and more symmetric. It would looks like other type of CMT. Some patients may also cause problem in their cranial nerves. For example, there are some patient had a hearing problem, and some patient may have feel a little bit of choking, and their muscle in their throat may be affected. There are other symptoms, and we'll start to gradually recognizing. We have studied and recently published a couple of years ago. We find the patient with HNPP have very severe fatigue, and we were really surprised because the degree of their fatigue even more severe than the patient with multiple sclerosis. That's something that was really surprising us. But there's a reason behind it. We can explain that in a few minutes. And then there's many patients who have CMT, HNPP. They have a problem with their GI function, and many of the patients end up diagnosis of irritable bowel disease. It is still something that is no clear answer yet. We're still trying to figure that out. If that's something that is caused by the HNPP mutation, or it's just a sort of association. We don't know the answer yet, but it's something that we observed quite frequently among the patients with HNPP. Pathologically, there's some degree of pathology in the myelin, and produce the structure called temacular. And as I showing here, and this is a nerve, the normal nerve, and here's a node of Ranvier where it produces the electrical signal. And then you can hear that's the area called perinodes. And in the HNPP patient, one of their perinodes end up pretty big, almost like swelled. And that's the increased myelination in that area, which we call temacular. That's what a temacular means. Okay. And there are also some patients who lost the axons, and particularly toward the later stage, both demyelination and axon degeneration did not happen until the later stage. And temacular happening in the younger age. But what we find is there's a lot of pathology that can happen very early, and that was not discovered until recent years by our laboratory. Next slide. So genetically, the HNPP is caused by missing of a gene. So and this gene have these DNA segments have total about 13 genes, and one of the gene called PNB22. So in the normal people, you have this area that has a whole bunch of genes in there. And then in HNPP, that's these whole segments of DNA is gone, called deletion. And that includes 13 genes, and one of the genes is called PNB22. So it's a very big piece of the DNA segments is missing. And this is very important to keep in mind because that gene piece is so big. So right now, all these gene therapy method and it's hard to be used for fix this disease because these are very, very different. Okay. Okay, go to next slides. All right, so what we find for the HNPP patient, before all kinds of pathology like temacular or other pathology happen, in their very early stage, they end up have a problem for myelin junction disruption. So these red dots are the myelin junctions. So in the middle, this is a nerve axon. It's almost like an electrical wire. And then you have insulating material around it. That's called myelin. The myelin wrapping around the axon many layers. But when you're wrapping around, each layer still have tiny small spaces. How would you seal that? Well, the nature designed have those proteins be made almost like a glue that glue them together, called myelin junctions, and seal them together so you will not leak those electrical current. But what we find what we found through our laboratory work, we find this junction got disrupted in the very early stage of the disease, almost like right after the born. And somehow that this junction disruption related to a molecular, next slide, related to a molecular called P21 activated kinase. And so that protein is a kinase. The kinase activity gets too high. So we designed a small molecular, and this is small molecular name. And then we block the kinase, and we were able to make the disease better. Why we know that better? Because this is the amplitude measurement for the electrical activity that usually gets blocked in HNPP. But once we give that medication, we can make the electrical current go reverse and go back to the normal. And then if we do that for a while, and if you don't do it and this electrical activity goes down during the aging, but if we give those medication to the animal model of the disease, we are able to block the decline of this problem, this physiological function. So it was successful in our preclinical study. And we are now in the stage of seek pharmaceutical partner to try to translate these treatment to the clinical for our patient. Next slide. So I think that I run out of time. I'm going to just wrap up with these final slides and to quickly mention that HNPP facing a very special challenge. In order to know that if our treatment is if successful or not, we need a good outcome measurement for the disease. But because the focality of these patients' symptoms, all other outcome measurement has been developed and may not be very well suitable for this disease. So we're actually now actively trying to develop a better outcome measurement for this disease, which is something that FDA really wants before we start clinical trial. So we're working on those. Next slide. I think that's it. So thank you for listening, and I'll be happy to answer any questions.
Thank you so much for that amazing presentation, Dr. Lee.